Bismillah, Assalamualaikum warahmatullah. You're listening to Immunology lesson on immunotolerance. Your goal in learning this topic is to understand what immunotolerance is and why it's important. And that's number one. And two, to discuss how it works among T and B cells. What immunotolerance means is the immune system's inability to respond to an antigen after being exposed to it. What we've learned in previous episodes so far is how the immunity responds to antigens. So here we learn how it avoids doing that. And a subset of that concept is self-tolerance, which is when our immune system tolerates or not responding to our self-antigens. Our immune system is trained to distinguish between self and non-self antigen. I mentioned this in previous episodes when I talked about the clonal deletion. Now, one of the reasons why it's important to learn this topic is that in some cases, the immune system may fail to recognize autologous antigen, which is another name for self-antigen, the antigens that originate from our own body. And that leads to an attack on healthy tissues, healthy organs. So this is called autoimmunity, and it results in autoimmune diseases. So these diseases can affect various parts of the body, including joints, skins, and other organs. To prevent harmful immune responses, your body has multiple mechanisms to regulate tolerance. And that includes the interactions between antigens and receptors, which can be influenced by the structure of the binding site, known as idiotype, and levels of certain immune molecules, such as cytokines like IL-2, and antibodies like IgG can also affect immune tolerance. And the third factor is certain types of immune cells, like regulatory T cells or Tregs, also play a role in promoting tolerance and preventing autoimmune diseases. Those mechanisms help inactivate lymphocytes that display high affinity receptors for self-antigens. What does it mean? It means if someone asks you, how come our immune system doesn't attack our own cells? The answer is self-tolerance. And if they ask, how does it work? The answer is our immune system has mechanisms to inactivate or turns off our lymphocytes that can hurt us. And we're talking about the ones that can bind strongly to our self-antigens. Self-tolerance can be developed in two sets of locations. So one is in generative lymphoid organs, such as the thymus and bone marrow. So there, the immature lymphocytes undergo central tolerance. So central tolerance means they learn to tolerate self-antigens before they mature. Secondly, self-tolerance can develop in peripheral locations. So example of peripheral locations are your secondary lymphoid organs, such as lymph nodes and spleen. There, the lymphocytes undergo peripheral tolerance. So peripheral tolerance means they learn the tolerance after they mature rather than before. So again, our immune system has tolerance mechanisms that prevent it from attacking our cells. So these mechanisms are called, number one, central tolerance, and number two, peripheral tolerance. So in central tolerance, you have immature lymphocytes that strongly react to self-antigens. So these guys may encounter the antigens in the thymus and bone marrow. So if that happens, they are either deleted, change their specificity, or develop into regulatory T cells. In peripheral tolerance, some self-reactive lymphocytes go away. They mature and enter the peripheral tissues. But in the peripheral tissue, they got evaluated again. So if they bind to self-antigens, they will be deleted or become anergic or unresponsive, or be suppressed by regulatory T cells. So we have two layers of safety mechanisms here. One is central, one is peripheral. So now let's take a closer look at the central T cell tolerance. Immature T cells in the thymus are constantly tested for their ability to recognize self-antigens with high affinity. Those that do recognize self-antigens with high affinity are eliminated to prevent autoimmune diseases. However, some immature CD4 positive T cells differentiate into regulatory T cells. So in our thymus, immature T cells are exposed to self-antigen. If the T cell recognizes a self-antigen, it will either die, we call it a negative selection or deletion, or develop into regulatory T cells, Tregs, which play a crucial role in preventing autoimmune diseases. So Imagine that this is an alien military recruitment camp where T cells learn to distinguish between self and non-self, between friend and foe. And those that fail to do it, they are either getting killed or they get taught to behave themselves differently. So that's what happened in central T cell tolerance. Now, in peripheral T cell tolerance, you have three mechanisms. You have energy, which means unresponsive. So energy occurs, for example, when lymphocyte encounters an antigen without co-stimulation. The second is suppression by Tregs, and the third is deletion. So a normal immune response involves the activation of T cells by antigen-presenting cells like dendritic cells, and that leads to T cell differentiation into effector cells and memory cells. If you can't remember what's going on here, watch the previous episodes on the adaptive immune response. Now, in contrast, peripheral tolerance prevents the activation of self-reactive T cells to avoid damage to healthy tissues. The mechanisms include, number one, energy, which makes the cell unresponsive. Number two, suppression by regulatory T cells or Tregs, where the Tregs block its activation. Or number three, deletion, which is through apoptosis, which is programmed cell death. So think of it like a self-destruct program. As I mentioned in previous episodes, when a CD4 positive T cell recognizes an antigen, it needs two signals to become activated. So the first signal comes from the interaction of the T cell receptor with the antigens. The second signal is provided by the interactions of co-stimulatory molecules. So without the second signal, the T cell becomes anergic, meaning it is unresponsive to subsequent stimulation by the same antigen. Again, the best way to understand energy is by comparing it with the normal T cell response. So when T cell recognize an antigen presented by an APC along with co-stimulators, T cell responses are induced. So if T cell recognize a self-antigen without co-stimulation, they become unresponsive. So this is called T cell energy. It happens because of a couple of possible factors. Firstly, there is a block in signaling from the T cell receptor or TCR complex. Or secondly, there is an engagement of inhibitory receptors. So in either case, the T cell becomes, the T cell remains viable, so it's not dead or anything, but it can't respond to self-antigen. So whether or not a T cell recognizes an antigen, it depends on a balancing act between activating and inhibitory receptor engagement. So this determines whether the T cell becomes activated or not. Inhibitory receptors like CTLA-4 and PD-1 play a crucial role in regulating T cell activation. So they act like brakes to prevent overstimulation of the immune system. For example, you've got your CTLA-4 on your Treg. So imagine you have an APC. The APC binds to Treg and to another T cell that is ready to activate. So in this situation, when CTLA-4 binds to APC, it signals the Treg to block the activation of the T cell. So that's the first thing. Secondly, CTLA-4 binds to B7 molecules on APC, right? When this happens, it removes the B7 from the surface of the APC. So imagine Treg keep pulling the B7 away from APC. Now, B7 is a co-stimulator on APC. What that means is T cell needs B7 to activate. So when B7 is no longer there, because Treg takes it away, T cell cannot activate. Another inhibitor, PD-1, is also a protein that can inhibit T cell activation. So it does that by blocking signals from T cell receptor, TCR, and co-receptor complex from co-stimulatory receptor CD28 and also other receptors as well. And this also results in T cell inactivation. All right, a lot is going on here, but just focus on this. Normally, T cell receptors, TCR, and co-stimulators like CD28, normally they can activate T cell. But sometimes you have PD-1. PD-1 is on T cells, yeah? So its ligands are on APCs. When PD-1 interacts with its ligands, such as PD-L1, it recruits a phosphatase called SHIP2. When this happens, SHIP2 inhibits signals from TCR and inhibits signals from CD28. So it does that by removing phosphate groups. But don't worry about that. Just remember that as a result of all this, T cell activation is suppressed. Regulatory T cells belong to the CD4 positive T cell subpopulation. Their main job is maintaining immune system balance by suppressing excessive immune responses. So these cells express high levels of CD25, which is a receptor for the cytokine IL-2. They also have a unique transcription factor called FOXP3. So the expressions of both CD25 and FOXP3 are crucial for the suppressive function of regulatory T cells. Regulatory T cells or Tregs can be generated by recognizing self-antigens in the thymus or in peripheral lymphoid organs. Tregs need FOXP3 and interleukin-2 as well, which is not shown here. They need both FOXP3 and interleukin for their development. In peripheral tissues, Tregs suppress T cell responses, like preventing naive T cell activation. They stop naive cells to become becoming effector cells. Tregs also inhibit other immune cells, such as NK cells and B cells, to make sure that they don't mount an overactive response that could lead to autoimmune diseases. Interleukin-2 or IL-2 is a signaling protein. IL-2 binds to receptors on Tregs that are recognizing the same antigen on the APC, such as dendritic cells. That binding of IL-2 allows the Treg to function. So in this way, IL-2 helps to promote the activity of Tregs. So that plays a key role in controlling the responses of T cells and preventing autoimmune diseases. So without the support of IL-2, Tregs would not be able to maintain proper immune balance and tolerance. Now, T cells that have a strong affinity for self-antigens, or ones that are frequently activated by antigens, are at risk of becoming harmful. So to prevent harm, the T cells undergo apoptosis, which occurs naturally in cells to maintain tissue homeostasis, meaning maintaining tissue internal balance. Apoptosis can happen through two main pathways: the mitochondrial or intrinsic pathway and the death receptor or extrinsic pathway. So there are two pathways of apoptosis. The first is the mitochondrial pathway. So this one is regulated by the BCL2 family of proteins. So this pathway is initiated by BH3 only proteins. So these proteins are induced because of cell injury. BH3 only proteins bind to BCL2 family effectors, which insert into the outer mitochondrial membrane. So what that means is when this binding happens, it makes the mitochondrial more permeable. That leads to mitochondrial contents like cytochrome C leaking out into the cytosol, and that activates the enzyme called caspases. So that activation of caspases leads to a series of steps like the activation of endonuclease and fragmentation of your subnuclear components and expressions of ligands which attract phagocytes. So all of these steps lead to death. The second pathway is called the death receptor pathway. Here, they use cell surface receptors. Examples of them are TNF receptors and FAS receptors. So when these death receptors engage their ligands, they activate proteins that assemble caspases. And like before, the activation of caspases leads to a series of steps and causes death. Regulatory T cells have several mechanisms of action that allow them to suppress the immune responses of other cells. So one way they do this is through CTLA-4 mediated inhibition of co-stimulation, like I explained before. By inhibiting co-stimulation, Tregs can prevent other T cells from becoming activated. So that's how these guys control the immune response. Tregs also produce immunosuppressive cytokines such as IL-10 and TGF-beta, which can inhibit the activity of other immune cells. Another way Tregs control the immune response is by absorbing IL-2, which is important for T cell proliferation. So when Tregs do that, they deprive other T cells of IL-2. So these various mechanisms allow Tregs to dampen the immune response and prevent excessive damage. All right, we're done with T cells. Now, for B cells, during their maturation process in the bone marrow, B cells first express a type of antigen receptor called IgM. So this IgM allows B cells to later bind to foreign molecules on viruses or bacteria or whatever. However, at this stage, B cells are considered functionally immature, meaning that they are not yet fully capable of mounting an effective immune response. So they're not there yet. Now, during this stage in the bone marrow, if these B cells identify self-antigens with high affinity, three different things may happen. One of them is receptor editing, where the B cells edits or modify its B cell receptor by gene rearrangement. Alternatively, if the B cell cannot undergo receptor editing, it may undergo deletion, a process where the B cell is eliminated from our cell repertoire, our cell collection. And finally, some self-reactive B cells may become anergic, meaning becomes functionally unresponsive. So during the maturation of B lymphocytes in the bone marrow, immature B cells first express IgM as their antigen receptor, but if they recognize self-antigens with high affinity or high avidity, doesn't matter, the B cells may undergo receptor editing, deletion, or anergy. Receptor editing involves the reactivation of certain genes. So that reactivation of genes starts a new round of recombination in the light chain of the B cell receptors. If you can't remember what the light chain is, go back to my previous episodes on antigen recognition receptors. But the basic idea is B cells can edit their dangerous receptors to become less dangerous to our own cells. Now, if editing doesn't work, B cells can undergo deletion, which involves apoptosis of the B cells that strongly recognize self-antigen. And alternatively, anergy can occur if B cells do recognize self-antigens, but the avidity is weaker. So what happens here is the downregulation of antigen receptor expression, meaning the B cell will have fewer receptors, or they just block the signaling altogether. Either way, they make the cells functionally unresponsive. Now, in addition to central tolerance mechanisms in the bone marrow, B cells that encounter self-antigens in the peripheral tissues can also undergo tolerance mechanisms, including anergy, deletion, and regulation through inhibitory receptors or through Tregs. All right, this is the last slide. So this is in the peripheral tissues, yeah? So B cells that encounter self-antigens can go through several mechanisms of peripheral tolerance. So one mechanism is anergy, where self-reactive B cells become unresponsive. Another mechanism is deletion, where self-reactive B cells undergo apoptotic death, which is death by apoptosis that I explained before. Another way tolerance can happen is through the engagement of inhibitory receptors. So it prevents B cell activation. So it basically sets a threshold for B cell activation that allows responses to foreign antigens, but not to self-antigens. And Tregs also regulate B cell activity, by the way. It's not shown here. So for example, when patients have defects in Tregs, you see increased numbers of what's called autoantibodies, so which are antibodies that attack friendly cells. All right, that's all. You can go over the video again the second time, but to summarize, our immune system is designed to tolerate self-antigens. So we call it immunotolerance. Immunotolerance involves the turning off or inactivation of T cells and B cells that recognize our own antigens. Central tolerance happens in the primary lymphoid organs, so the bone marrow for B cells and the thymus for T cells. And then you've got your peripheral tolerance, which occur in secondary lymphoid organs and other tissues as well. So in both stages, lymphocytes undergo negative selections to eliminate the ones that recognize self-antigens. So this negative selection process involves things like receptor editing, deletion, or energy, depending on the stage of development and the degree of self-reactivity. So that's the big picture. So if you like what you're learning here, I encourage you to go beyond the surface explanation that I gave you in this episode, because we can use our understanding of immunotolerance to develop therapies for autoimmune diseases and other conditions where the immune system is dysregulated. All right, that's all. Until next time, keep on reading, keep on learning. Barakallahu li wa lakum. Wassalamu alaikum warahmatullah.